We are going to continue with the fun, with continuing with our Max Weber lecture series. We are very happy to have with us today Anna Dreber from the Stockholm School of Economics. And I'm going to pass on the floor to Alexandra Jabbour, who will be telling a bit more about our speak. Please.
Thank you, Juho. So it's a great pleasure for the UI community and me to welcome Professor Anna Dreber for the first Max Weber lecture of 2025. Professor Anna Dreber completed her graduate studies at the Stockholm School of Economics in Sweden. She is the Johan Björkman economic professor at the Stockholm School of Economics. Additionally, she is the Wallenberg Scholar, a member of the Royal Swedish Academy of Sciences, and a member of the Royal Swedish Academy of Engineering Sciences. Professor Dreber is affiliated with the Department of Economics at the University of Innsbruck in Austria, and the Credence Goods, Incentives and Behavior Group, an incubator for collaborative and transparent economic sciences. Her research focuses on behavioral and experimental economics and the meta-science, focusing on the credibility and reliability of scientific results. She conducts applications, setups prediction markets, and organizes forecasting surveys to assess whether researchers can predict replication outcomes, as well as the results of new hypothesis tests. Her work is interdisciplinary. She's collaborating with researchers in psychology, anthropology, medicine, and neuroscience. Professor Dreber is one of the leading social scientists working to improve the credibility and reliability of scientific results. She has conducted replications in laboratory experiments in economics, published in Science, and in psychology, also published in Science. Furthermore, she has published research on predicting replications outcomes with papers in PNAS and Nature Human Behavior. Professor Dreber is also involved with the Institute for Replication, which organizes replication games across the globe. Additionally, she is engaged with the Lab Square, a hub for meta-science research with a specific focus on economics. So without further ado, please join me in welcoming Professor Anna Dreber. We look forward to a lecture followed by questions from the audience. Thank you.
Thank you so much. Thanks for the invitation. Thank you for coming. I'm very much looking forward to interacting with you. So I'm going to talk for 45 to 50 minutes, and then let's have a conversation. And I have too many slides. We don't have to go through them all, but let's get started. Okay, so I'm going to start with a phenomena that maybe I should stop using as an example, but I'm still not ready, and I will explain why. So this is a phenomena called power posing. Has anyone here not heard about power posing? A few people. Okay, so power posing is based on a study published in 2010 in a top psychology journal, Psychological Science. So in this study, they report that they have 42 participants, so 42 participants who are randomized to sit or stand like this for a minute or two, or sit or stand like this, basically an expansive pose or a contractive pose. And in this paper, they argue that if you sit or stand like this, you get higher testosterone levels, lower cortisol levels, so lower stress hormones, you become more financially risk-taking, and you have higher feelings of power. And that's just completely amazing that from this very tiny intervention, you get these dramatic hormonal and behavioral effects, right? So this has led to what is now the third most viewed TED Talk with about 75 million views. And as long as sort of the number of views keep increasing in the millions, I think I'm going to continue using this as an example. And why? So you see the risk-taking result from the original paper on the slide. Those in the high power condition are statistically significantly more risk-taking than those in the low power condition. So the problem here, and sort of, we were not out to get power posing in any way. We actually believed in power posing. So that tells you something about maybe my naive beliefs. So you should maybe discount some of what I say, given my bad judgment here. But so when we set out to replicate this, because we wanted to extend it, because we thought it was very interesting with power posing, maybe it can help us close a gender gap in risk taking or something. This is what we find. We basically find nothing. Here's our risk-taking result, 5 times the sample. We make the study experimental blind, unlike the original study. We find nothing on testosterone, nothing on cortisol, nothing on risk-taking. We do find the effects on feelings of power that many studies since then have failed to replicate. Basically, it looks like nothing is going on for power posing. So the good news about this example is that the world of academic psychology / behavioral science has moved on. Most people don't believe in power posing anymore. The first author on the paper has written an open letter saying sort of what they did that was a bit problematic and why you should basically forget about power posing. So I think that's extremely honest and good, and we learn we can make mistakes and we move on. But the rest of the world continues loving power posing. So that's why I'm going to stick with this example. It's very hard to correct a false positive result like this once it's been published and once it's out sort of in the TED sphere. So that's why I'm still hung up on this case. But we'll come back to this. Okay. So another title of today's talk could have been the more informative but slightly longer version, how there have been or are replication problems and how results might not generalize, and how we could design studies and do pre-analysis plans and multiverse analysis and change incentives and explore heterogeneity and do team science, etc., to solve this problem. So I think there are some problems. There's not one silver bullet to this. I think how you're going to solve whatever replication problem you have in your field will depend on the methods you use, the type of data you use, etc. I'm mainly going to talk about experiments, but that's because I come from experimental economics, so I do experiments. But this is, of course, not just an experimental problem. The beauty of experiments is that if you do them in the lab, you typically can replicate them. You can collect new data and test these studies again, or if you do them online, as you do. So sort of the fact that we can replicate things in the lab doesn't, and sometimes unsuccessfully, doesn't mean that you should think that results are necessarily more credible for non-experimental data. This is, of course, not just a social science problem, even though in our research we have focused on social science studies. There is unfortunately lots of evidence suggesting that problems may be worse in some parts of medicine where we could think that stakes are maybe higher. But I think to a large extent, we have an incentive problem. The way we do research, even though most of us are here because we're interested in finding out how something works, it's easy to fool yourself and others, and it can be wasteful. And I think sort of with our scientific methods, eventually we will weed out false positive results, but I think we can speed up this process. And if you disagree or think otherwise, I mean, let's have a conversation. Okay, so which scientific results can we trust? We know that that's going to depend on many different things. Like what's the p-value of the original study? What statistical power did the original study have? It depends on whether you are engaging in any of the so-called researcher degrees of freedom. Are you phishing? Are you p-hacking? Are you forking? So a p-value is not just sort of some type of false positive probability. That depends on how you calculated it, right? We also know that it's going to depend on publication bias. Most journals in many fields, at least, are more likely to publish positive results than null results. Knowing this, many authors, researchers choose to not even write up null results, so they remain in the file drawer. So if we only read published results, of course, that can be misleading in terms of what do we actually know about some scientific question. So which results can we trust will also depend on things like priors. So what's the probability that the hypothesis is true in the first place? So when we do frequentist statistics, this is not something that we explicitly incorporate, but I think some of us do think about priors anyway when we read papers. I mean, if I say that I would do a study testing the hypothesis that people are more likely to sleep lying down than standing up, I think many of you would assign that the prior close to 1. Whereas if I'm testing whether water has memory, and which would break a chemical laws, so many of you would probably assign that the prior closer to 0. So why don't we incorporate priors more when we think about evaluating research results? Typically because they're subjective. I mean, we could have different priors for power posing. We probably did, and I had a very naive prior. But they're also hard to access. So if I would like to access your priors to evaluate something, how should I do it? And then we've been arguing that maybe with the help of surveys and prediction or decision markers, we can get to priors and think about how to use them when evaluating results. Okay, so before talking about some results of others that have failed to replicate, I want to be the first one to sort of mention that I have results that I think do not replicate. And here's one that has been haunting me for many years. So in this study, this is something I did as a PhD student with co-authors in anthropology and psychology. We were interested in exploring genetic variation in risk-taking. So we looked at two specific dopamine receptor genes. So dopamine is the reward system in the brain. So it's not completely crazy to think that variation in dopamine-related genes could predict variation in risk-taking. So we do a study with about 100 men. We take a DNA sample of these men and we genotype these samples for two specific dopamine receptor genes. This was at the time when it was expensive to genotype, thus only 2 genes. We find that those with the longer version of one gene here, DRD4, are statistically significantly more risk-taking than those with the shorter version of the gene. That's the result you see. We find a null result for the other gene, DRD2, and we report that in the paper. It's not in the headline, and I don't even think it's in the abstract, but it is in the paper, so we do not hide that this null result, okay? So what's the problem here? What's not to like? We basically have 0 statistical power. Even if you're trying to be Bayesian, there's nothing to learn from this type of study. Why? A sample size of 100 is just completely meaningless given what we now know from genome-wide association studies. I didn't know that at the time. I was super enthusiastic about this study. I spent so much money, time, energy, years digging further into this, doing another study with maybe a little bit larger sample, couldn't find any main effect, but in some subsample somewhere, there was always something statistically significant. That was just complete noise and such a waste of time. Have I retracted the paper? No, there's no fraud, error. I just don't believe in it. So it's not that easy to correct this sort of result once it's out there, but it's easy to make mistakes, and I wish I had learned faster. Okay, so I'm going to talk about a bunch of different papers and projects in a short amount of time. If you're interested in any of this, just send me an email and we can discuss it more, and I can send you a paper or whatever you want there. So I'm going to talk about replications, and we typically like to differentiate between so-called direct replications and conceptual replications. In a direct replication, we try to run basically the original study in a new sample. We try to stay as close as possible to the original study that we can, ideally using the same methods and materials as the original authors, but obviously we do it in a new sample. In a conceptual replication, the idea is instead to vary something either in the design or in the tests, and then see sort of do the results replicate. Personally, I think conceptual replications are more interesting than direct replications, but I typically want to start with a direct replication. If the result does not replicate them, I'll probably stop. If it does replicate, then it's interesting to think about conceptual replications and generalizability. So it's, I mean, now I say this as if it's super clear what's a direct and what's a conceptual replication, and the lines can be blurry and subject to lots of discussion. So we typically say that a result replicates if we, in the new data, the replication data, find an effect that is in the same direction as the original study, and this effect is statistically significant in the two-sided test, p less than 0.05. Many people really dislike this binary way of taking a continuous variable. We also discuss almost as much relative effect sizes. So what is the replication's effect size compared to the original effect size? And as I will show you, when we have high power in the replications, these two measures correlate very well. There are also many other interesting replication indicators that I will not go over because there won't be enough time. So I'll talk about some of these projects, some of the ones that we did many years ago and some that are more recent. So I'll give you an overview of these results. I'll also talk about work that we have done using prediction markets and decision markets and surveys to predict the outcomes of these replication projects. I will talk about some work that we're also doing on sort of analytical heterogeneity, design heterogeneity, that basically puts limits on how much you learn from one specific study. If there is time, which there probably won't be, I will talk about selective reporting of placebo tests. But instead of talking about that later, let me just spend 30 seconds on it. I mean, it's hard to assess the credibility of non-experimental results because we cannot typically replicate those studies. We cannot get new data. We can do robustness tests. I think we can do multiverse analysis and other very interesting things. We can also look at sort of the data a bit like from a like more general perspective. So what we do in a recently published paper is that we look at selective reporting of placebo tests. So placebo tests are big in economics. I think they're getting bigger in political science. These are basically tests that are not supposed to be statistically significant. Nothing is supposed to be going on in these placebo tests, and that's support for the research design that you have in a natural experiment or something else. So we search a bunch of economics journals for a longer period of time and just count the number of statistically significant placebo tests. If these are true null hypotheses that are being tested, 5% of them should be statistically significant by chance, but they are not. So half of them that are supposed to be there are basically missing. So that tells you that we have some problems also with placebo tests. And then I'm not going to talk more about that. But placebo tests can be great, but right now I think they can be a bit problematic. All of this is, of course, joint work with tons of people, and I'm not going to name them all because that would take the rest of the 30 minutes that we have. Okay, so some older replication projects before we get to the newer ones. So for me, like it all started with the replication of power posing, plus, my own failed study of genetics and this project in psychology. So this was a big, ambitious replication project led by Brian Nosek and many other researchers. At the end of the day, there were about 270 authors on the paper that resulted from this project. I played a very minor role. So the goal was to try to replicate as many studies as possible from 3 top psychology journals published in 2008. At the end of the project deadline, 100 studies had been replicated. Among the 100 original studies, 97 reported statistically significant results. So you can see the original studies to the left in the figure. Among the 100 replications, nothing magical is happening at p less than 0.05. About a third of the results replicated. So this then led to headlines saying there's a replication crisis in psychology, and I think that's sort of a correct conclusion to draw. But maybe the replication rate is actually slightly better than what people, including myself, considered at the time. So power was supposed to be high in these replications, in the sense that power is about 90% to detect 100% of the original effect size. This sounds great if we think that original effect sizes are not inflated. And we have reasons to believe that they are, and I'll come back to this. So very much inspired by that project, together with co-authors, we set up a related project in economics, where we went through 2 top econ journals during a specific time period and searched for all experiments in the lab, testing a specific hypothesis with students or other accessible subject pools. At the end of the day, there were 18 such studies. We perform high-powered replications, meaning we have 90% power to detect 100% of the original effect size. And we find that 11 out of 18 results replicate. So we were a bit worried about this power issue, whether we were actually underpowered. So in the next project that we did, again with many co-authors, we decided to try to replicate social science studies published in Nature and Science, specific time period, 2010 to 2015, using students or other accessible subject pools. So we had 21 studies that we attempted to replicate. Here we do have higher power. So we do potentially a two-stage procedure. So the first round of data collection, when we replicate the study, we have 90% power to detect 75% of the original effect size. If the results do not replicate, we do a second round of data collection, so that we, in the pooled data, have 90% power to detect 50% of the original effect size. So we allow true positive results to also have been inflated when they were first published. And at the end of the day, when we have such higher power, we find that 13 out of 21 results replicate. Relative effect sizes for these 13 out of 21 studies are about 75%. So even true positive results tend to be inflated. Relative effect sizes for the 8 studies that do not replicate according to this binary statistical significance criterion are about 0%. So these 2 measures correlate well. Okay, so to these studies, we added prediction markets. Maybe some of the economists and political scientists know what prediction markets. I don't know to what extent everyone else is interested. But anyway, so prediction markets have been used a lot to study sort of political outcomes, sports outcomes, and other things. Typically in these markets, you have people betting on binary events. Will Obama become president in 2008? Yes or no? A contract in Obama is worth $1 if Obama becomes president, and $0 if he does not become president. So the sort of why prediction markets could be good to aggregate information would be because they're not about my political preferences, they're about my beliefs. So if I do a poll when I ask, and so I want to do a poll and figure out who will people vote for, I typically need a large representative sample of voters who will truthfully reveal their preferences. All of those things can be difficult to achieve. Here I can have a weird self-selected sample of participants who just bet here according to their beliefs. And I could potentially aggregate a lot more information from this type of sample, because how I bet here will be a function of what do I read, what do my friends and family say, who I might not agree with when it comes to politics, but there's potentially better ways to get information here. So instead of having people bet on who will become president, we ask researchers to bet on which studies will replicate. So a hypothesis here is worth $1 if the study replicates and $0 if it does not. And replication here means that we find an effect in the same direction as the original study, and this effect is statistically significant in a two-sided test. If sort of you think the probability is higher than the current market price, you should go long in this contract, you should buy it. If you think the probability is lower, you should short sell this contract. So we invite participants when we set up these markets. They're typically economists, psychologists, behavioral scientists. We give them money to trade with, between $50 and $100 depending on our budget. And we use a logarithmic scoring rule so that there's always a counterparty to trade with. We are that counterparty. So we want to interpret the price of these contracts as the probability of the study replicating according to these markets. So prices start at 50 and can potentially go up or down depending on how people are betting on these studies. So we have these markets open for about 2 weeks, and depending on the markets, we have between 30 and 200 participants. So who are these participants? PhD students, faculty, researchers who are interested either in replications or prediction markets. We don't know much more about them, but this is what they are. Before they participate, we of course inform them about the studies that we're trying to replicate. We inform them about the original studies, about the replications, including replication reports, where we tell them how we selected a specific result, how we did our power calculation to justify sample size, etc., and how our study differs from the original study, because it always differs, if not in timing, in sample or something. Before participants, researchers come to these markets, we ask them in a survey to give us the probability of each study replicating, and then they can enter the markets. So we've done this now for a bunch of different replication projects. So a subset of the big psychology project, we did for all the experimental economics and the nature and science replications, and for 2 of the so-called many labs projects in psychology. So right now we have 123 studies for which we have a replication outcome, prediction market prices, and survey results. So this is sort of not much in the sense that I think about power and importance of statistical power, and now I'm showing you 123 studies, so we can't say that much, but we can at least say something. So pooling these results, and so we say that if prediction market prices or survey beliefs are above 50, the markets or the surveys think that the study will replicate. So in black dots here, circles, we have successful replication outcomes, and in non-solid circles, we have unsuccessful replication outcomes. And you can see that there are more black or solid dots above than below 50. So there is sort of some wisdom of crowds, but it's far from perfect. So how well do prediction markets perform? Okay. And it's also a bit hard to generalize any result here, in the sense that if we find that they work out really well in some context, That's going to be a function of what are the studies people are trying to predict, what are we replicating, and who chooses to participate in the markets. And the latter is something that we cannot control necessarily. We can think that we've set up a perfect prediction market project and nobody cares. But then we're not going to learn much from it. Okay, but I'm going to a bit self-servingly zoom in on the markets where it worked the best, where we also had the highest power in our replications. And that's the social science replication projects. This is where we replicated studies in nature and science. So, and had the high statistical power. So in blue here, we have successful replication outcomes, and in yellow, we have unsuccessful replication outcomes. In diamonds, we have prediction market prices, and in circles, we have survey beliefs. And you can see that all blue diamonds are to the right of all yellow diamonds. So this is almost too good to be true. So they worked out extremely well here. So why is this interesting? Not clear. I mean, I'm interested in this. I don't ask you to necessarily think that this is so interesting. I think this tells us something about that there is something systematic about results that successfully versus unsuccessfully replicate. And researchers are pretty good at picking this up. In these markets, you can also learn something about the beliefs of others by seeing how they are betting. So you can be more Bayesian about it. And that's potentially why markets could perform better than surveys in predicting results. We can also use these prediction market prices to back out priors about the tested hypothesis in different stages of the testing process, which I also think it's interesting from like a philosophy of science perspective. Okay, so there's also some work, and I played a very minor role in this Altmade et al. Paper that by Adam Altmade. But there's work on machine learning models, trying to sort of see whether using statistics of the original studies, using like words, word length, etc., of original studies, you can predict replication outcomes. So in these projects, the machine learning models are compared to the prediction market results. And so far, the markets have performed as well or better than the models. That's going to change, I think, with better, more sophisticated machine learning models and more training data, which is happening. Okay, so there was another project that I'm going to be very brief about. So we tried to scale this to not just 20 studies that we were replicating and having people predict, but 3,000 studies where a subset would actually be replicated, but we wanted prediction market beliefs for all of these 3,000 studies. That was not that easy to get, because who wants to bet on so many studies? So this, yeah, it's hard to scale. The biggest problem here was just that we had very few participants, so very little trading, and this is still a work in progress. So based on sort of combining this interest in replications and prediction markets, we're thinking, can we use the markets for something else, helping us decide how to move forward? There are many studies that could potentially be replicated. How should we choose them? I mean, we can define some criteria, we can pick the ones that we are the most interested in, or have some type of voting, or use some other type of mechanism. So we decided to use decision markets instead. So decision markets function similarly to prediction markets in many ways, but now we let market prices help us move forward, basically. So in a decision markets, all studies that could potentially be replicated must have a positive probability of being picked for replication. And then we have to scale payoffs according to their probability of being picked for incentive compatibility. But we can put extra weight on certain studies depending on our decision rule. So I might think that the studies that are the most interesting to replicate are the ones where the information value of replication would be the highest, so where prices are closest to 50. Or I might have some other decision rule, like I want to put extra weight on studies that have the highest chances of being false positives or true positives if I want to move forward and design interventions that people really think will work, or something else. So we recently we worked on this for many years, but it was recently published, did a sort of what we think is a proof of concept paper on using decision markets to help us select which studies to replicate. So in this project, we have a detailed pre-analysis plan that is followed unless I mention a deviation, just make that clear. And this is, again, it's joined with many people including Felix Holzmeister, Magnus Hannesson, who played a very important role. Okay, so we decided to sort of select a larger set of studies and then to replicate the smaller sets. And large and small are not that different in this particular case. So we went through PNAS, the Proceedings of the National Academy of Sciences, USA, that journal, time period 2015 to 2018, searched for all social science experiments that had been done online on a platform called Amazon Mechanical Turk. We searched for all between or within subject treatment designs that were logistically feasible. I mean, starting this, I thought that everything would be logistically feasible if it's run online, but the 3 turned out to be just impossible for us to code for various reasons. And these studies had to have at least one statistically significant main or interaction effect in the main text. Many of these studies or papers could have several studies that had been run on MTurk. When that was the case, we decided to randomly pick a study. So in the big psychology project, the last study was typically picked for application because that's supposed to be like the voila results of the paper. In the Nature and Science replication project, we picked the first study because that's typically the one that is presented as the most important. Both of those methods for choosing a study was criticized. So here we said that, okay, randomness should at least be the best to pick a study. That was, of course, also criticized. So whatever, I mean, you can never make people completely happy. So at the end of the day, we have one study from each of these papers that we're trying to replicate. To what extent does this give us something generalizable for these papers? Very unclear, but this is what we did. So with these criteria, we ended up with a study of, at the end of the day, with 41 papers that we potentially could replicate. But we did not want to replicate all of them. We wanted to replicate 26 out of these studies. So we ran decision markets for 41 studies. So the decision markets here function similarly to the prediction markets. We ask researchers to spend time thinking about these studies. We give them money that they can use them to bet on the studies. And we said we will replicate the 12 studies that have the lowest prices, the 12 studies that have the highest prices. So the studies that the markets think are the least and most likely to replicate, and 2 random studies among the remaining ones. And we scale payoffs according to the probability of being picked for replication so that everything is incentive compatible. Okay, why only 26? I mean, ideally, we would have had 400 papers and would have said we're going to replicate 250 of them. For budget reasons, that was not the case. But also think about being a researcher here who is trying to predict things. Just going through 41 replication reports is quite some work. And then thinking, okay, what probability do I assign each of these results replicating versus doing that for 400? So that's why we, I think, do a somewhat underpowered proof of concept, but we had to do something. Okay, so we invited researchers mainly through economics and psychology email lists. We have 162 social scientists that thankfully participated. They're given a survey before the markets and they get detailed replication reports about what we are doing in our replications. We also share these reports with the original authors for feedback. And they typically reply. If not when I write them an email, then when my more senior co-author, Colin Cameron, writes them an email, then they reply. Okay, so in this replication, we basically use the same sampled restrictions and exclusion criteria as original studies. So we have 90% power to detect 2/3 of the original effect size in a 2-sided test. So at the end of the day, our average sample size in our replications is about 3.5 times as large as what the original studies had. So we were 5 teams who did these replications. We had attention checks and IP quality checks before random assignment to treatments. I mean, many people who do online experiments are worried about who are the participants. Are they real? Are they bots? How much attention are they paying? So that was also one of our worries, obviously. Mistakes were made. I can spend the rest of the time talking about them, but I won't because I want to get to the results. So what are the results? Here we have the 41 studies. In green, we have successful replication outcomes. In purple, we have unsuccessful replication outcomes. And if there is no color, that means that there is no replication. these studies were not selected for replication. So we find the positive correlation between prices and replication outcomes, R of 0.5 Pearson correlation. P is equal to 0.008. We would call that suggestive evidence and not statistically significant, because we keep P less than 0.005 for statistically significant. When we compare sort of the top 12 with the bottom 12, we find some suggestive evidence suggesting that replication rates differ from between the top and bottom 12. So some evidence suggesting that decision markets can be used as a tool, but not perfect evidence suggesting this. It's also, I think, interesting to note that average relative effect sizes are overall about 45%, but zooming in on those that replicate versus not replicate, according to this binary statistical significance rule, we find that average relative effect size is about 70% for those that are statistically significant in replications and 3% for the ones that fail to replicate. So again, these measures correlate pretty well. So what have we learned from this? I think, as I mentioned earlier, original studies tend to overestimate the effect sizes of true positives. So if we do meta-analysis, and I love meta-analysis, we should be a bit worried about true results being overestimated on average. There is also something systematic about results that fail to replicate. Maybe we can use decision markets more. Original p-values are relevant. They are pretty good at predicting whether a result will replicate. This applies to these studies that are from a specific point of time, of course. So it's not clear how to think about this for newer studies where I think people zoom in on p-values for good reasons to a larger extent. But I mean, if you read a study with testing a very surprising hypothesis with a p-value close to 0.05 in a small sample, chances are pretty high that the result will not replicate. Okay, I will skip this. So now I have maybe 5 minutes or 10 minutes. So then I'm going to get a little bit to analytical and design heterogeneity. So I think one big lesson for many people, including myself, from these projects was that pre-analysis plans are great. If you do an experiment, why not pre-register it properly with a pre-analysis plan, saying how you would collect the data, how you would treat the observations, whether you will exclude anyone for some reason, exactly what hypothesis you're testing, what are your primary hypothesis, secondary hypothesis, exploratory hypothesis, and potentially later non-pre-registered exploratory hypothesis, and you say exactly how these tests will be done. So you can basically write the code before you run the experiment. Things are, I think, wonderful in many ways because you get credible p-values and you don't end up fooling yourself, which is so easy to do. And of course, you can do exploratory tests. It should just be transparent what is confirmatory versus what is exploratory. I think lots of the results that turn out not to replicate in the original studies were exploratory, but were presented as confirmatory. So the pre-analysis plan will show sort of one potential fork in the garden of forking paths. If you can look at the data in many different ways, you tie your hands to one specific path when you write the pre-analysis plan, and you get meaningful p-values. But potentially there are many forks possible to explore, and depending on who I ask to design a study and write the pre-analysis plan, I can get different pre-analysis plans and different forks, right? So this wouldn't result in any systematic bias in effect sizes, but could underestimate the standard error in statistical tests. So sort of lead to this non-standard standard error that would need to be incorporated. So then the question is, of course, how large is the variation in results depending on who you ask? Sort of, is this something that we need to worry about or not? And there are different ways to explore this. I mean, there's the so-called multiverse or vibration of effects or specification curve analysis approach, basically looking at what is the theoretically justified set of analysis and results in a given data set. And here, I think it's interesting to note that researchers in psychology have been talking about this at the same time as sociology, psychology, medicine, political science are basically all doing different types of multiverse analysis, saying that when you can test things in so many different ways, there is room to pick a specific specification that can be very meaningless. So instead, show everything. So another way, instead of looking at sort of what's the theoretically justified set of analysis and results, you can give the data set to different researchers and ask them to test a bunch of hypotheses on the same data and see what's the natural variation in analysis and results in a given data set. So this approach is called the multi-analyst or many-analyst approach. And the first paper that I know of that published on this was a paper by Silbersan, Ullman, and Nosek with many co-authors, where they gave the same soccer data to a bunch of different researchers and asked them to test the same hypothesis and got some variation in results. When this was first presented, headlines were like, oh, so much variation depending on who you ask. Now I think this is actually the least variation that we've seen from a multi-analyst project. So we have I will talk about a couple of ones that I've been involved in, but there are many others. So here's a study we did in sort of neuroeconomics, neuroscience, where we gave the same fMRI data sets to a bunch of different research teams and asked them to test the same 9 directional hypotheses based on previous studies using a similar mixed gambles task. And by we, I mean my co-authors who are neuroscientists who did most of this work. So our main outcome variable that we're interested in here is basically the fraction of teams reporting a statistically significant result, yes or no, based on their own criteria for each of these 9 hypotheses. So here are the 9 hypotheses. You can sort of read what they are. Maybe someone knows some neuroscience and can infer something. Otherwise, you can just look at this other column, the fraction of teams reporting a statistically significant result. If there would be complete consensus, we would be close to 0 or 1 for this variable. And we are fairly close to 0 or 1 for 4 out of 9 hypotheses. And for 5, we're somewhere between chaos and consensus. And it's interesting to note that for each of these 9 hypotheses, there's at least one team that finds it's a statistically significant result, and it's not the same team. So think about a world with publication bias. You could see all of these 9 hypotheses being published with support, whereas in some cases, a majority of researchers would not have found something for the same data. So what did this lead to? Lots of discussions in neuroscience on like how is data being processed and analyzed? Why are people making these different decisions for the same data set, testing the same fairly clear hypothesis? So we decided to do something similar for financial data. So we have 164 research teams testing the same 6 non-directional hypotheses using very sort of big data from the German stock exchange. This was led by Albert Menkveld in Amsterdam. So here we sort of gave them the data and the hypothesis and asked them to write up short reports explaining what they did and what they found. These reports were then given to a group of senior researchers in finance who wrote basically like peer reports, peer reviews of these reports and feedback on how they could change things. So this review element decreased the variation in results, but there were still tons of variation in what people found. So in these figures, you have the reported estimates on the in the top figure and then reported t-values. So depending on who you ask, you really can get statistically significant results that are massively different in different directions by very good researchers. So we can zoom in on those that have top publications in finance or economics, and there's still tons of variation in what they find for the same data and same hypothesis. So that's sort of about analytical heterogeneity. So we talked earlier a little bit about how do we think about replication when we cannot get new data? If you do lab experiments, you can get new data. If you do field experiments, maybe you can get new data. Most of social sciences that are trying to do quantitative work cannot get new data. How do we evaluate the credibility of such results? I think these many analysts or multiverse approaches are one way forward to explore a bit sort of what is there room to find. You can do a multiverse analysis and plot the distribution of results so that the reader can see, are they, by chance, there should be some more results and some in the opposite direction, but are most of them in the hypothesized direction, yes or no? So that's sort of analytical heterogeneity. The other thing would be design heterogeneity. So how much variation is there in results depending on who you ask to design a study. And that's something we explored for sort of the fairly big question, does competition affect moral behavior? What do we mean by competition? You choose. What do we mean by moral behavior? You choose. We said that we will pay for experiments that you design, you meaning researchers, that will be run online on the platform called Prolific. They should take a maximum of 15 minutes, which is fairly standard for such experiments. They should use monetary incentives. There should be a control and a treatment, 200 participants in each. And that's it, basically. Then you can choose how to define these different things. Competition can mean markets. They can mean competitive tournaments, payment schemes, many different things. Moral behavior could be generosity, could be dishonesty, public goods contributions, your call. So the idea here was to basically say, like, for this type of research question, what variation in results can we get when we try to keep the analysis constant and have constraints on the designs in some ways, but not other ways? And this was very much inspired by another project led by Justin Landry and Eric Ullman that I played a very small role in, where the analysis was not really kept constant, but the goal was to explore design heterogeneity somewhat relatedly. Okay, so we invited researchers. When you do this type of sort of crowd science, you never know if anyone would sign up. So luckily people were interested in this. So we had more research teams signing up than we expected. We thought that maybe 20 teams would sign up. So we had funding for that. 100 teams signed up. We didn't have funding for that, but we got extra funding so we could actually have designs from 50 research teams that were randomly picked. And we used their designs and implement them all at the same time online, 18,000 participants. And then we run the same analysis for all of them and want to see what is the variation results. So I'm going to be super brief here because time is running out. If you focus on the right part of the figure here, B, that's a meta-analytic. These are the meta-analytic results for the 45 different designs that we at the end of the day have. So in yellow or orange at the end of the figure, there's the overall meta-analytic effect size, which in this particular case is small, but statistically significant and negative. Okay, so given our constraints, we find a small but negative effect of competition on moral behavior. But there is also quite some variation depending on the design. And this, I would argue, even though there are many null results here, maybe because we have pre-analysis plans for all of these studies, is that there's actually quite a substantial design heterogeneity. So it's 1.6 times as large as the average standard error of effect size estimates. So when we start combining these different sources of heterogeneity, what do we have? So we have population heterogeneity. That's something most of us have been thinking about for quite some time. So we and others would argue that there's also design heterogeneity to consider and analytical heterogeneity. So what do we do with all of this? We need to do something about confidence intervals and p-values, and things are not going to get easier, unfortunately. So we have a paper that was recently published on this in case anyone is interested in this. Okay, so I think maybe last slide. So now I've been talking a lot about experiments. And as I said, that's because I'm an experimental economist. But there are really no reasons to believe that things are better, at least in other parts of economics. I cannot talk about sociology or very little about political science. But there are a few very nice papers by Abel Brodeur and co-authors where they look for evidence of p-hacking in different subsets of economics. And I know that others had done this earlier in political science. So they basically look at the distribution of p-values or c-values, and then they can transform them to p-values. Distribution of c-values for different literatures. What is the true distribution like? Unclear. But if there are sort of missing c-values corresponding to specific p-value thresholds, things look a bit weird. So here, basically, when you see two peaks in a valley, that means that some p-values are missing and typically show up just below statistically significant, p less than 0.05. And they find evidence for this for papers that have so-called stars, eye catchers in tables versus but also for papers that do not have stars. I don't think stars are the problem. I mean, they find this for papers that have theoretical models, as well as for papers that do not have theoretical models. They basically find these weird 2 peaks in a valley in all subsets that they look at for 3 top econ journals, a longer time period, but the category lab experiments or RCT data, where we also know that there are problems. So this is sort of, just because you can't see evidence of this in the distribution doesn't mean that there are no problems. But this, I think, is a nice way to look at sort of, is something weird going on in a literature? Okay, I think that was it. At the end of the day, I think all of this results sort of in maybe us potentially shifting significance thresholds to p less than 0.005 rather than p less than 0.05. This is one of the most hated, I think, ideas that I've been part of, and this is from a paper where we're, I think, 72 authors. I think only these 72 authors like this idea, and the rest of the world do not. So we can discuss this if you have nothing else that you want to say later, or read the paper. But lots of collaborators here. Thanks so much to everyone. If you want to talk about this later, just send me an email and I'm super happy to talk about this. Thanks.